Hi, Assalamualaikum warahmatullah. My name is Shakila, and in this video we are going to talk about the experimental analysis. So in order you to do a scientific experiment, you have to keep two things in your mind, which are the groups and the variables. So now let's talk about the group first. There are two different groups in any experiment. The first one, we call it as control group. It is used for comparison to the other group. So the other group, we call it as experimental group. So the control and experimental group only differ by one variable so that you can measure what you want to know in your experimental group. And another thing you need to keep in your mind is that the sample size. If you are handling with human or animal study, you need to use a large sample size, at least 30 sample size. And another important thing that you need to remember is a trial. Trials refer to the replicate groups that are exposed to the same condition in an experiment. For example, John is going to test each sugar variable 3 times. Let's give an example. You are going to do a scientific experiment comparing two different groups of plant. Each of the group at least to have a minimum 2 plants per group. If you have more than 2 plants, it's even better. The control group contain 2 plants that grow under natural sunlight, while the experimental group contains 2 plants that grow under red wavelength. So the purpose of this experiment is to determine the difference of plant growth between 2 types of light. So we call this experiment as a prediction test. A prediction must be tested to, number 1, actual experiment, which is from your experimental group, and second is control experiment, which is from your control group. Alright, we've done on groups, so now let's talk about the variables. There are 3 important variables. Number 1, independent variables. Independent variables are the thing that being manipulated or changed between the control and experimental group. For example, the type of plant that the type of light that used for plant growth. Second is dependent variables. Dependent variables are the things that changes as a result of the experiment. For example, at the end of the experiment, what you want to measure is the amount of plant growth. Maybe you want to look at the height of the stem of the plant, or maybe number of the leaf. Alright, so third variable is the control variable. What is control variable? It is the factor or condition that kept constant, so they don't affect the result. For example, you want to measure plant growth using 2 different types of light, and you use the same amount of water and the same type of soil for both control and experimental group. Alright, next, after you've done your experiment and you get the data, so you can do next is the analysis of your experiment. So experimental analysis could be done after the data collection, but maybe you are facing some problem during analyzing data. What are the problems? Maybe number 1, sampling error. For example, you cannot examine every subject in a big population. I give you an example. I give a situation. Let's say you want to determine obesity among generation Gen Z in Malaysian population. So how do you sure that you get every sample in each region in Malaysia? So you only study a sample or subset of them to represent the whole population. And other than that, you can use statistical analysis. For example, ANOVA test or t-test or correlation to reduce your sampling error. Another problem during analyzing data is equipment error. Okay, after you analyze your results, you need to report your finding. So please present report according to the analyzed data and results. And your result must contain introduction, literature review, description of methods, results, discussion, conclusion, as well as the references, and publish finding in scientific journals or present to the audience so that you get comments and suggestion for improving your experiment.